Folate's contribution to fertility remains one of its most underappreciated roles, quietly influencing reproductive potential in both men and women through mechanisms that go far beyond its traditional association with pregnancy support. While folate is widely known for preventing neural tube defects in the developing fetus, emerging research reveals that it also plays a foundational role in gamete health, conception success, and early embryonic development. These effects are often invisible until they manifest as unexplained infertility, recurrent miscarriage, or suboptimal outcomes in assisted reproductive technologies. What makes folate's role so intriguing is that it influences fertility not only structurally, through DNA synthesis and repair, but epigenetically, through methylation pathways that determine the functional quality of sperm and oocytes. In women, folate is crucial for the maturation of oocytes and the proper development of the uterine environment. Adequate folate supports DNA replication during the rapid cell divisions of oocyte maturation and early embryogenesis, while also ensuring that methylation marks are correctly established and maintained. These epigenetic signatures govern gene expression in the embryo and play a vital role in implantation, placental development, and embryonic viability. Women with folate deficiency may have eggs of lower chromosomal integrity, which can result in reduced fertilization rates or an increased risk of miscarriage. Moreover, the uterine lining's receptivity is also influenced by methylation activity, and folate deficiency may compromise this critical interface, making conception less likely or more prone to failure. What often escapes mainstream discussion is the parallel role that folate plays in male fertility. Folate is essential for spermatogenesis, the process by which sperm are produced, and for maintaining the stability of the sperm genome. Studies have shown that men with lower folate intake or blood levels tend to have a higher incidence of sperm DNA damage, lower sperm count, and reduced motility. These defects are not merely quantitative but also qualitative, affecting the ability of sperm to fertilize an egg and produce a viable embryo. One of the key pathways at work here is DNA methylation. Sperm DNA must undergo precise epigenetic reprogramming, and folate serves as the methyl donor in these reactions. When folate is insufficient, aberrant methylation patterns can emerge, potentially disrupting gene expression in ways that impair embryo development or lead to poor outcomes in vitro fertilization, IVF. The presence of MTHFR gene polymorphisms adds a further layer of complexity. This genetic variation impairs the body's ability to convert folate into its active form, 5-methyltetrahydrofolate, which is needed for proper methylation. Individuals carrying these variants may appear to have sufficient folate on lab tests, but still suffer from functional deficiency at the cellular level. In men, MTHFR mutations have been associated with impaired spermatogenesis and abnormal sperm methylation profiles. In women, these variants may contribute to higher rates of miscarriage and implantation failure. The implication is that folate status cannot be reduced to mere intake. It also involves genetic capacity for utilization, making folate a prime candidate for precision nutrition in fertility treatment. Another hidden facet is folate's role in reducing oxidative stress within reproductive tissues. Homocysteine, when not properly metabolized due to folate insufficiency, can accumulate and generate oxidative damage. This is particularly detrimental in the delicate environment of the reproductive tract, where oxidative stress can impair egg and sperm quality, disrupt hormonal signaling, and interfere with embryo implantation. By facilitating the conversion of homocysteine to methionine, folate helps maintain redox balance and preserve the integrity of gametes. This makes folate not only a builder of DNA, but a protector of the biochemical environment required for successful conception. Folate's subtle influence extends into assisted reproductive technologies as well. Couples undergoing IVF or ICSI, intracytoplasmic sperm injection, may benefit from optimizing folate status prior to procedures. Higher folate levels in women have been associated with improved ovarian response, better embryo quality, and higher implantation and live birth rates. In men, folate supplementation has been linked to improvements in sperm count and motility, potentially boosting the success of fertilization even in cases of male factor infertility. These findings suggest that folate is not just a supportive nutrient, but a modifiable factor that can tip the odds in favor of conception when other variables are held constant. Ultimately, folate's connection to fertility is both profound and multifaceted. It safeguards DNA integrity, supports essential methylation pathways, defends against oxidative stress, and quietly governs the success of life's earliest and most fragile steps. Its deficiency may not always produce immediate or dramatic symptoms, but its absence can create hidden roadblocks that frustrate the most intimate human endeavor, reproduction. In recognizing folate as a cornerstone of reproductive health, we also open a door to new possibilities in diagnosing, preventing, and overcoming infertility, often with nothing more complex than correcting a nutrient imbalance.